For 70 years, scientists believed the secret of Albert Einstein's genius was locked inside his brain. They sliced it into 240 pieces, mounted it on microscope slides, measured every fold, counted every cell, photographed it from angles the man himself never imagined. And after 7 decades of study, they found nothing definitive. But there was something else hidden in Einstein's biology that nobody thought to examine for decades. Not in the shape of his cortex or the density of his neurons, but deep inside his chromosomes, in a code that had been passed father to son for over 12,000 years. The code connects the 20th century's greatest physicist to a people who may have changed the course of human civilization itself. The story begins the way most Einstein stories do. In Princeton. On the morning of April 18, 1955, Albert Einstein was dead. A ruptured abdominal aortic aneurysm. He was 76. He had asked to be cremated, his ashes scattered privately. No worship, no relics. But the pathologist on duty at Princeton Hospital, a Yale-educated doctor named Thomas Harvey, had other ideas. During the autopsy, Harvey removed Einstein's brain. No one had authorized this. Not the family, not Einstein. Harvey simply took it. When Einstein's son Hans Albert discovered what had happened, Harvey persuaded him to allow the brain to be kept, for science. The condition was clear. Results published only in reputable journals. No spectacle. Harvey weighed the brain at 1,230 grams. That is well within the normal human range. No obvious sign of genius there. He photographed it from every angle, then transported it to the University of Pennsylvania, where a technician spent 8 months sectioning it into blocks, embedding them in celloidin, and producing 12 complete sets of microscope slides. And then, nothing happened. For years, nothing happened. Harvey lost his position at Princeton Hospital in 1960. He drifted. Kansas, Missouri, Wichita, Titusville. He carried the brain with him in mason jars, in cardboard boxes, once reportedly in a cider crate stashed beneath a beer cooler. He lost 3 marriages, lost his medical license in 1988 after failing a competency exam. He ended up working night shifts at a plastics factory in Lawrence, Kansas, living in a cramped apartment above a gas station. His neighbor was the beat poet William Burroughs, who reportedly joked he could have a piece of Einstein anytime he wanted. The most important brain of the 20th century, stored in a basement by a man the scientific establishment had forgotten. It was not until 1985, 30 years after Einstein's death, that the first study was published. Marion Diamond, a neuroanatomist at the University of California, Berkeley, reported that certain regions of Einstein's brain had a higher ratio of glial cells to neurons compared to 11 male control brains. The finding made headlines. But skeptics pointed out the control brains averaged 64 years of age, while Einstein's brain was 76, and glial cells naturally increase with age. Later studies highlighted unusual features in his parietal lobes, an absent groove called the Sylvian fissure, an extra ridge in his frontal lobe. Each finding was announced with fanfare, and each was followed by caution. As mathematician Brian Burrell put it, 6 different reports on Einstein's brain, each highlighting a different anatomical feature as the possible source of his brilliance, have collectively built what one critic called a neuromythology of genius. Here is what nobody seems to have noticed while they were counting cells. The entire search was built on an assumption. That genius was structural, visible, that if you zoomed in close enough to the tissue, you would see intelligence written into the architecture. But the brain is not a building. It is a process. And the tools Harvey used in 1955, formalin preservation, celloidin embedding, Nissl staining, these were designed to reveal anatomy. They could show you the shape of a neuron. They could not tell you what that neuron knew, or how it got there, or what deep lineage shaped the biology of the man who carried it. For that, you needed a completely different kind of evidence. And it would not emerge from a mason jar in someone's basement. It would emerge from a laboratory decades later, using a technology Harvey could not have imagined. The answer, when it came, did not arrive from a microscope slide. It arrived from a different kind of laboratory entirely, one that reads code instead of shape. And it did not require Einstein's brain at all. It required a living relative and a single swab of saliva. Here is how it works. Every human male inherits his Y chromosome from his father, who inherited it from his father, in an unbroken chain stretching back through thousands of generations. Unlike most DNA, the Y chromosome passes with almost no shuffling or recombination. Mutations accumulate slowly, like notches carved into a walking stick passed from father to son across centuries. By reading those mutations, geneticists can assign a man to a haplogroup, a branch on the vast family tree of all human males. And crucially, they can trace where that branch originated and when. Now, Einstein himself was cremated. No intact DNA remains from his body, but Y chromosomes do not require the original carrier. Any male descendant in the direct paternal line carries essentially the same genetic signature. A man identified as a patrilineal descendant of Naphtali Hirsch Einstein, born in 1733, who was Albert Einstein's great-great-grandfather, provided a DNA sample. The result was haplogroup EZ830, a subclade of E1b1b, one of the most geographically far-reaching lineages in the human male family tree. Before we see what this connection reveals, if you are finding this discovery as fascinating as we are, subscribe so you do not miss future stories like this one. Now, EZ830. Most people hearing that designation would have no reason to pause. It is a technical label, one branch among thousands on the Y chromosome tree. But in 2016, a landmark study changed its meaning entirely. Iosif Lazaridis and an international team of geneticists published a paper in Nature analyzing ancient DNA from 44 individuals across the Near East, some dating back 12,000 years. Among them were 6 Natufian skeletal remains excavated from Raqefet Cave in what is now northern Israel. The Natufians, a people most of the world has never heard of. And yet, by any measure, they are among the most consequential populations in human history. Living between roughly 12,500 BC and 9,500 BC, the Natufians were among the first humans anywhere on Earth to abandon nomadic life. While the rest of humanity still followed herds and seasons, the Natufians settled. They built stone dwellings with foundations. They harvested wild cereals with composite flint sickles. They ground grain with heavy stone mortars and pestles, tools that could not be easily carried, evidence of people who had chosen to stay. They buried their dead with shell ornaments and carved bone figurines. They created art, engraved stones, decorated objects that suggest symbolic thought. They are credited with founding Jericho, one of the oldest continuously inhabited cities in the world. The Levant in their time was not the arid landscape of today, but a verdant woodland rich with over 100 kinds of edible plants, cereals, fruits, and nuts. And in the DNA extracted from 3 of those ancient Raqefet cave skeletons, the geneticists found a paternal haplogroup, EZ830, the same lineage that runs through the Einstein family. Let that settle for a moment. The man who reimagined the universe, who bent space and time with equations, who gave humanity E equals mc squared, carries in his Y chromosome, a direct paternal thread connecting him to the people who first bent the arc of civilization itself. The people who looked at wild wheat and imagined cultivation, who looked at temporary camps and imagined permanent homes. 3 things make this connection credible and not merely poetic. First, what the evidence shows. EZ830 is not a common haplogroup. It is a specific, well-defined branch. The EM35 phylogeny project has identified 4 distinct clusters of EZ830 carriers in the modern world. 2 of those 4 clusters are exclusively Ashkenazi Jewish in origin. Einstein's family were secular Ashkenazi Jews from the Swabian region of southern Germany, tracing back through generations of merchants and tradespeople in towns like Buchau and Jebenhausen. The haplogroup fits precisely within the broader pattern of Ashkenazi Jewish paternal lineages, which are predominantly Near Eastern in origin, with less than 0.5% admixture per generation over roughly 80 generations. Second, what it rules out. This is not coincidence, and it is not a statistical fluke. In 2014, a team led by Shai Karmi at Columbia University sequenced the genomes of 128 Ashkenazi Jews and determined that the entire modern Ashkenazi population, some 10 million people, descends from a founding group of approximately 350 individuals who lived 600 to 800 years ago. Every Ashkenazi Jew alive today is, at most, a 30th cousin of every other Ashkenazi Jew, but the paternal lineages within that population trace much further back. A study by Hammer and colleagues, published in the Proceedings of the National Academy of Sciences, confirmed that Ashkenazi paternal chromosomes share deep common ancestry with other Jewish populations and with non-Jewish Middle Eastern groups, including Palestinians and Syrians. The Y chromosome does not lie. It is a receipt carried across millennia. Third, what it implies. Einstein's paternal lineage does not begin in medieval Germany or Roman Palestine or even the Iron Age kingdoms of Israel. It reaches back to the Natufians, to the Epipaleolithic Levant, to a moment when humanity itself was pivoting from one mode of existence to another. The broader parent haplogroup, E1b1b, is estimated to have originated in East Africa roughly 22,400 years ago. From there, it moved northward into the Horn of Africa, across the Sinai, and into the Levant. The Natufians carried it. The first farmers of the southern Levant carried it. EZ830 has been found in 75% of the Ain Ghazal Neolithic population near modern Amman, Jordan, dating to approximately 8,800 BC. It persisted through the pre-pottery Neolithic, through the Bronze Age, through the rise of Semitic language communities. And somehow, carried by a chain of anonymous fathers, it survived the Babylonian exile, the Roman destruction, the medieval pogroms, the bottleneck of 350 souls, the migrations across Europe, and arrived in Ulm, Germany in 1879 in the body of a baby boy named Albert. But this story is not settled. It is important to note that the International Society of Genetic Genealogy classifies the Einstein haplogroup attribution as debatable. The DNA sample came from a man claiming descent from Naftali Hirsch Einstein, Albert's great-great-grandfather, but the full genealogical chain connecting the tested individual to Albert Einstein has not been published in a peer-reviewed journal with the level of documentation some geneticists would prefer. The finding is consistent with what we know about Ashkenazi Jewish paternal lineages, but it is not confirmed in the way, say, the Richard III identification was confirmed through multiple independent lines of evidence. There is also a broader, still active debate about what Ashkenazi ancestry really means. For decades, the prevailing model held that Ashkenazi paternal lines were Near Eastern, while maternal lines were substantially European, suggesting that Jewish men migrated to Europe and married local women. But a 2025 study by Livni and Skorecki challenged this view directly, analyzing mitochondrial DNA and arguing that both maternal and paternal lineages trace to a unified founding population from the Near East. If confirmed, this would mean the Ashkenazi community was far more genetically cohesive from its origins than previously assumed. Other researchers remain cautious. The question of exactly where and how the founding population mixed remains open. What is not debated is the deep Near Eastern anchor of the paternal lineages. That signal is consistent across multiple studies, multiple labs, multiple decades. And then there is this. Einstein is not alone in his haplogroup. The broader E1b1b lineage includes Napoleon Bonaparte, the Wright brothers, and President Lyndon Johnson. It is the most common Y chromosome lineage in North Africa, reaching 80% prevalence in some Berber populations. It is found across the Horn of Africa, in the Mediterranean, the Balkans, and scattered throughout Europe. In 2018, ancient DNA from Morocco's Grotta des Pigeons, dating back 15,000 years, revealed the same parent lineage. It is a haplogroup that moves, a lineage of people who crossed continents and, wherever they arrived, reshaped what they found. Today, 46 thin slices of Albert Einstein's brain sit behind glass at the Mutter Museum in Philadelphia, stained, mounted, labeled. Visitors lean close, peer through the microscope eyepieces, searching for the spark. But the real story was never in the folds. It was written in a code far older than the brain itself, a paternal line stretching back through 12,000 years of anonymous fathers, to a hillside in the Levant where a people nobody remembers decided to stop wandering and start building. They did not leave monuments. They left something more durable. A genetic signature, passed quietly from father to son, across empires and exiles and bottlenecks that should have erased it. It survived anyway. If Einstein's Y chromosome genuinely traces to the Natufians, the people who first planted seeds and imagined a different world, what does it mean that the same lineage produced a man who planted ideas that reshaped our understanding of the universe itself? If this hidden chapter of history intrigued you, subscribe to explore more mysteries the evidence is only now revealing.